Welcome to The Explainer. Today, we're doing something incredibly cool. We are breaking down Dr. Chem's stunning experimental demonstration to completely demystify the ultimate blueprint of life, and we're going to do it using standard household items. We're taking a highly cinematic journey today, going straight from the macro world of your everyday kitchen counter all the way down into the microscopic, glowing architecture of a plant cell.
I want you to start by just thinking about this for a second. DNA is packed so incredibly tightly into microscopic cells that it seems practically impossible to see without a high-powered electron microscope. I mean, a single strand of DNA is roughly 35,000 times thinner than an average human hair. There's no way you can just look at it, right? Well, today, we're basically going to pull off a molecular heist. We are going to extract millions of these invisible strands and physically force them to clump together until they become totally visible to the naked eye.
Okay, so let's dive into this. The answer to how we actually pull this off starts with a simple, everyday fruit. Now you might be wondering, out of everything in the grocery store, why a banana? Well, while humans are diploids, which means we have 2 sets of chromosomes, bananas are actually triploids. They have 3 sets of chromosomes crammed into their cells. This makes them the absolute perfect candidate for yielding a massive, highly visible amount of DNA. Plus, their soft texture makes breaking them down incredibly easy for us.
All right, let's move into Section 1, the Kitchen Science Toolkit.
Here is our physical toolkit for the experiment. But I really want you to look at these not just as basic baking ingredients, but as highly specific molecular instruments. In this explainer, the dish soap is going to act like a pair of microscopic scissors. The table salt, that'll act like a magnetic shield. And that ice-cold alcohol is going to be the grand stage where our invisible DNA finally makes its dramatic appearance. Just keep one thing in mind. This is a real science experiment, not a smoothie recipe. So please, whatever you do, do not drink it.
Moving right along to Section 2, breaking down the barriers.
So, before we start aggressively mixing our chemicals, we really have to define exactly what we are trying to achieve here. The main goal is a biological process called lysis. In biology, lysis literally means to break open the plant cells. It's a breach. We have to shatter the sturdy outer cell wall, completely dissolve the flexible phospholipid membrane, and finally pop open the nucleus itself because that is where the genetic gold is actually hidden away.
Here's our physical protocol. Consider this our master roadmap for the lab. It gives us a really clear structure to track exactly where we are in the experiment at all times. First, we physically disrupt the tissue. Then we chemically dissolve the cellular barriers. Next, we filter out the garbage. After that, we precipitate the DNA. And finally, we collect it. Let's look closely at that very first step.
Notice the physical action you see happening here, vigorously mashing the banana. As I mentioned, you're not just making baby food here. If we were to digitally zoom in to the microscopic level, you would see that you are physically tearing apart the rigid cell walls. These walls are incredibly sturdy, almost like a thick paper bag protecting the delicate contents inside. By mechanically disrupting the tissue until it reaches a smooth, pudding-like consistency, you completely expose those inner membranes to our upcoming chemical attack.
Next up, look at the physical action of stirring in the soap. On a chemical level, what's actually happening is the soap is ruthlessly attacking the phospholipid membranes. It's exactly like how dish soap cuts through tough grease in your frying pan. The soap acts like a molecular wedge. It literally slips between the lipids and dissolves the fatty membrane that surrounds the cell, like a stretchy rubber glove. And when that tiny molecular envelope finally pops, the DNA spills right out into the liquid. But we hit a bit of a roadblock. DNA molecules have a negatively charged phosphate backbone, which means they naturally repel each other. To visualize this, just imagine the heavy, stubborn resistance you feel when you try to push the 2 south poles of a pair of magnets together. They just bounce off each other, right? Well, this is exactly where our table salt comes to the rescue. The positively charged sodium ions in the salt act like a third magnet slipped right between those repelling poles. It perfectly neutralizes the resistance, so the millions of DNA strands can comfortably aggregate and clump together.
Which brings us to Section 3, filtering the genetic soup. As you observe the physical filtering happening here, the underlying chemical reality is that we are trapping all the cellular garbage. By simply pouring our mashed mixture through an everyday coffee filter, we effectively separate the genetic gold from the biological trash. The filter successfully traps the unbroken cells, the torn walls, and all the large proteins. What gently drips through into your glass below is a perfectly clear liquid called the filtrate. In this liquid, the DNA is completely freed, it's perfectly intact, but it's still invisibly dissolved in the water.
All right, Section 4, the magic of precipitation.
We have officially arrived at the climax of the experiment. To get the DNA to finally solidify and show itself to us, we have to force it into a state of precipitation. Right now, the DNA is happily dissolved in our salty, soapy water. We need to drastically and violently change its environment so that it can no longer stay dissolved.
See, the thing you need to know is that DNA is heavily hydrophilic, meaning it absolutely loves water. Because of this, it hides perfectly in our aqueous solution. But DNA hates, and I mean absolutely despises, alcohol, particularly chilling, ice-cold ethanol. When it comes into contact with alcohol, its molecular structure can no longer remain dissolved.
Notice the extremely careful pouring technique here. The cold alcohol suddenly drops what we call the dielectric constant of the solution. If you look closely at that exact boundary where the two liquids meet, bam! Millions of DNA molecules instantly clump together, precipitate completely out of the solution, and rise up like these incredible cloudy thread-like strings. You can literally take a wooden skewer, reach right into the glass, and spool the blueprint of life directly onto the stick. It's totally wild to see.
And finally, Section 5, From Banana to Biotechnology.
So, you've just successfully spooled DNA onto a wooden stick using stuff from your kitchen. But what I really want you to take away from this is that the exact same basic principles, lysis, filtration, and precipitation, are used literally every single day in high-tech biotechnology labs across the globe. Whether a forensic scientist is isolating DNA from a microscopic drop of blood at a crime scene, or agricultural researchers are developing new disease-resistant crops, the foundational chemistry remains exactly the same as what you just did.
It's absolutely foundational. All of modern genetics fundamentally starts with this beautiful, simple chemical separation. You just cannot sequence a genome, cure a genetic disease, or understand our ancient human ancestry without first successfully pulling that DNA out of the cell so you can actually work with it in the real world.
Our whole world is built on these invisible instructions, and today, you learned exactly how to bring them out into the light. If a bit of soap, salt, and alcohol can reveal the complex genetic code of a banana, what other microscopic secrets do you think are hiding in the strawberries, kiwis, or even the vegetables sitting on your kitchen counter right now? Keep exploring, keep examining the world like a scientist, and I'll see you in the next explainer.